Researchers at the Sainte-Justine Hospital are testing what might be a major breakthrough in helping children with heart defects. They're using a 3D printer to make heart valves.
Dr. Savoji, a researcher with a notable background in chemistry and biomedical sciences, has recently published an article on the formulation and evaluation of PVA, gelatin, carrageenan inks for 3D printing and development of tissue-engineered heart valves. This breakthrough offers the possibility of customizing heart valves for each patient, contrasting with current methods that rely on animal-derived or mechanical valves and need patients to take anticoagulation medication for a long time, which can lead to severe complications. In this video, Nika Sajudi Asli, will interview Dr. Savoji to get more familiar with him and use his experiences in his life as a researcher. Dr. Savoji, thank you for accepting my invitation to this interview. Your academic journey is impressive. Could you share more about your academic background with us?
My academic journey began with a focus on chemical and process engineering in Iran. During my studies, I became fascinated with biomedical engineering, leading to my first master's degree in this field. At that time, the field was just starting to gain attention. I worked on a drug delivery system project in collaboration with a pharmaceutical company, developing and fabricating a transdermal patch for nitroglycerin for people with cardiovascular disease. This system was designed for sustained release, administering medication through the skin over 7 days. Subsequently, I worked in the industry for 5 years while pursuing my PhD in chemical engineering in Iran. I then moved to Canada, where I spent a year as a researcher at the University of Ottawa, working on a project for Environment Canada focused on water purification. Following this, I decided to return to my interest in biomedical engineering and pursued my second PhD at Polytechnique Montreal, specializing in the fabrication and characterization of vascular grafts using the electrospinning technique. I created small vascular grafts with diameters of less than 6 millimeters and conducted their mechanical, physical, and morphological characterizations. Afterward, I joined the University of Toronto for a 3-year postdoctoral research position, where I started working with 3D printing. It was there that I familiarized myself with tissue engineering and organ-on-a-chip technology. Eventually, I received an offer from the University of Montreal, where I moved to establish my own lab.
As you mentioned, you have your lab, Savoji Lab. Could you tell us more about your lab and the work you do there?
I established my lab at Sainte-Justine Research Centre in 2020. Our focus is on biofabrication and functional biomaterials, including bioprinting, 3D printing, lithography, soft lithography, microfabrication, nanofabrication, and organ-on-a-chip for drug testing and disease modeling. And our 2 main pillars are macro tissues and tissue regeneration for implantation purposes.
You have experience conducting research in both university labs and hospital research center labs. Could you tell us about the main differences between these 2 environments and the potential advantages and disadvantages of having a lab at a hospital's research center?
I believe there are more pros than cons. Being in a research center positions you in a real-world setting where you can identify the actual problems that need solutions. Some of the doctors are also researchers, with labs at the same center. This creates excellent opportunities for bioengineers to become more involved in the field and fosters great collaborations. A challenge, however, is the language barrier between engineers and the clinical sector. But with experience, this issue tends to resolve itself, and both parties become more motivated to interact with each other.
Throughout your research career, including in your current and future projects, there's a notable focus on cardiac tissues. Is there a specific reason or personal motivation behind your emphasis on this area of research?
Statistically, cardiovascular diseases affect 1/3 of the population, with cancer impacting another third. Historically, cardiovascular issues have been prevalent, emphasizing their importance and commonality, and this prevalence was the main reason behind my focus on cardiac tissues.
Producing an organ can help many people in the world, especially a heart, which is more challenging to find a donor, but still there are many limitations to reach to this point. In your opinion, how far are we from producing a functional heart?
The heart, due to its complex physiology, pathophysiology, genetics, and engineering aspects, is one of the most challenging organs to engineer. Currently, we are far from creating an artificial living heart because of numerous challenges, such as producing big number of cells in the lab or even in bioreactors. We still have a long way to go.
Sometimes in life we wish that we had a magical power to overcome the problems and limitations. As an experienced researcher in biofabrication and biomedical fields, if you had this magic power, what would have been the things that you would have changed or enhanced to accelerate organs and tissues bioprinting?
First, all equipment should be in a good manufacturing practice lab, so my first wish would be to create many GMP labs for this purpose. Then, we will need numerous bioreactors to produce billions of cells to be able to create an organ. Identifying the best materials to use is also a question that needs to be answered. For my fourth magic, I would minimize human interaction and increase automation for the entire process. Finally, I would wish for accessible printers with higher resolution and precision.
You have been in academia for many years, and you have been a part of many projects during these years. Among these projects that you have been involved in, which one is your favorite, or the one you are most proud of?
My latest research, which took about 3 years, is my favorite. It involves printing functional heart valves for both pediatric and adult patients using a new biocompatible ink made of PVA, gelatin, carrageenan, with no cytotoxicity and immune response. This print has been done without any support or bath, and we used extrusion-based printers for them. The valves were responsive under physiological, aortic, or pulmonary conditions without any overlap. We've conducted small animal tests and plan to focus on this area for the next few years.
In about an hour, this machine will reproduce something so precious it has the potential to add years to a life, and it's roughly the size of a raspberry.
I really believe this is really promising.
PhD student
Because,
Armin Jafari and Sainte-Justine Hospital researcher Houman Savoji found a new way to make heart valves. Using hydrogels, known for being compatible with tissue generation, the valve is produced in a 3D printer. Stem cells could then be harvested from the patient, put on the valve, and grown in a bioreactor before being implanted into the patient.
That's incredible. We previously talked about the importance of understanding the real problems to solve them. Beyond the innovative aspects of this project, what is the key reason that makes this study so important in the real world?
Current market valves are made of metal or part polymers and are fixed in size. When we implant them in pediatric patients, they can't grow with the patient and require replacement every 5 to 10 years, meaning multiple open heart surgeries. With our growing valve, the need for repeated surgeries is greatly reduced.
Every research project in our field follows a long path with many steps towards commercialization. For this specific project, what are the next steps that you have to work on?
We have plans to test it on medium-sized animals like mini pigs, examining functionality and modeling by creating a flap in the aorta. Then, we'll move on to larger animal models, with sheep as the ideal candidates for heart valve implants.
In life, progression paths are often nonlinear and unpredictable, and things don't always go as planned. Is this also true in the context of research?
In reality, about 99% of our efforts result in failure, with only 1% leading to success. Accepting this fact is going to help us. These failures provide invaluable experience, feedback, and information, helping us refine our methods and achieve success.
As the last question, if I ask you to give 3 pieces of advice to biomedical students to help them in conducting better research and achieving greater success in their field, what would those recommendations be?
Firstly, interact with experienced individuals and learn from them. They have first-hand experiences that are so valuable. Step out of your comfort zone and attend diverse conferences even if they are not exactly in your fields. Secondly, take advantage of all the possibilities of learning new things in different fields. This can be on scientific subjects or even on subjects like project management, time management, organizational skills, and entrepreneurship. This broad perspective is invaluable. Lastly, be passionate about your work. If you are not passionate about it, you should find the subject that interests you.
Thank you, Dr. Savoji, for generously sharing your time and insights with us. I also extend a big thanks to everyone who is watching this video. And to all of you, I hope for great success in your research and in your lives.